basically, so 1 year ago, we were talking about what is bioinformatics, etc. And another challenge that we have been having during this project is about, okay, we want this data to be out in the public and other researchers to make use of it, and how we do that. And then we talk about repositories, where yes, where it's not possible, etc. So that was a suggestion that maybe it would be good to have this type of masterclass where we discuss on data repositories and what it means for science, and especially for decision. So we have prepared like 3 steps or 3 parts of the presentation. The first one, we will talk about history and a bit of overview of databases repositories over the last 70 years. Then Christina will talk about database repositories in decision. And then Ferran will talk about, okay, once you have the data, cooking the data in the good sense, right? Doing a proper analysis of it. So if we think about, okay, the history of databases in the context of bioinformatics, the first thing that I would like to clarify or to talk about is that within most of the definition of bioinformatics, it's included the aspect of collection, storing, analyzing, and disseminating data. And this is important because we generate some data set, we do the analysis, and maybe some other researchers will make an additional use of this data in other contexts. Or we pull different data types and make another thing, right? But we need to make it in the proper way. So when I was saying 70 years, I was not exaggerating. So I would like to ask if anyone recognizes this name. Because that was from the masterclass from the last year. Okay. So Nuria specifically made that point, right? We are having her here. I will talk why we are talking about Margaret today. She was generating this atlas of protein sequence and structure. It was clear that at that time there was a development where they were able to sequence proteins, identifying amino acid, and that was this collection of books, right? And she was one of the leading forces there. Next slide is actually from Nuria from the last year, when she was saying, well, that was a development, and then there was a challenge associated to this development, because the proteins were not fully profiled, were pieces of it, right? So if you have a full protein and you have pieces of it, you need to reconstruct it from all these small pieces that you have, right? And that was the first kind of bioinformatic effort. And the comprotein, which was the generation identification of these full protein based on their pieces. And that's why Margaret was so important in this context. So that was also another slide from Nuria. And from that time, we have been seeing a lot of growth on many different aspects. And I want you to, guide or discuss about it. And this is not by any means, it's a full, complete description. We only have 10, 15 minutes, but it raises some question that is important for us. After proteins, the next that was quite important was the nucleotide databases. And it's interesting because Margaret was a mathematician. One of the ones who developed the first nucleotide databases was a nuclear physicist in Los Alamos that identified the need to make these collections. So this is one moment where you see like people from other fields are recognizing some needs because they have this, maybe looking into the data, we need to have the data, and they created the sequence database, then it transformed into the gene bank. Importantly, it's open access, annotated, so nucleotide sequences and the protein translations. And first collections, CD collections, now we can download them, right? But at the time, for some time, you even have tapes moving this collection from place to place. United States, it's happened. In Europe, we have the EMBL data library along the same times. Then Japan, And then basically, this is something that is quite recurrent on the nucleotide databases and the proteins, etc. US, Europe, and Japan has this kind of international consortium making all these collections. What was important was, okay, you create a repository. And the idea of the repository is you have identified a new nucleotide, please share it with the community. This is important, but not everyone is enforced to do that. It's a recommendation. The point is, it became enforced in some of the journals to make it happen. And this is when really things are starting to grow. And this is something that is recurrent in most of the data repositories. It's not only to have the repository there, it's actually to enforce through the publications, through the journals, enforce this requirement for publication. With databases, we have similar kind of developments. I think it's really interesting if you take a look. SwissProt was generated by this Amos Byrock. He has this paper kind of a number of years later. He was a person that wanted to do computation and wanted to be on the lab and identify the need of having this collection of proteins to a different level and showing like, okay, not only just the sequence, but also the function, 3D conformation, variants, etc. Extremely interesting read for anyone who is interested in the history of how these things tend to happen. There were other developments, and you can see also structural databases about the 3D conformation, etc. But databases are not only about proteins or nucleotide sequences, etc. Sometimes we have functional annotations. That's also another type of database. So if you have identified a gene associated to a pathway in a given, and then you publish this, should be one trying to collect these pathway gene associations. And it started at some point with the KEGG. That was kind of in Japan. You have seen these plots in the presentations before today. And we have these databases on different years growing. And you can see that basically we are ending using the same ones right now. So this has been evolving, better annotated, etc. This is interesting, but a major change happened when you have the genomic era, right? The whole human genome for the first time sequence. Massive project, lasted for quite a number of years. It was finished 2 years ahead of time, which is quite rare, right, in research. And once you have these millions of bases, right, you want to visualize them, but not only visualize the sequence, you want to have a notation track, like where are the genes, where are the different isoforms, where are the SNPs, etc. And that required to have visualization efforts. And this is where you have the Ensemble Visualizer, UCSC Genome Browser, which is in itself already a database for all these resources and how to interact with them. Okay, that made a change. But the next change that also happened and made a major impact to every one of us was the high throughput. And right now, when we think about high throughput, many times we think about sequencing, but actually the first label for high throughput was macroarrays, where you can, using fluorescence levels, you have kind of barcodes for each one of the genes, and then you are profiling. Fine, I'm not going to go into the details. But the good thing is through this, we have genome-wide a scale of gene expression, then SNPs, then DNA methylation. And it started again. You have one group generating a macroarray data set. Fine. They publish the data. Someone wants to access this data again. How do you do that? You have to put a repository out there. You have a link. So again, we have Gene Expression Omnibus, a repository. This is the first paper with the first design, all these acronyms. Anyone that has gone into Gene Expression Omnibus knows them from by heart, because you have the different profiles of the arrays, the different dataset collections, series, etc. That made a major impact. What is the problem? One thing is to have the repository there. The other thing is enforcing everyone to upload the data to the repositories. And well, before I go into that detail, this is something quite interesting because you can see the amount of expression that has been growing. These databases are growing and growing and growing. But another revolution that happened is you can see the amount of expression by array is being replaced slowly, but continuously by next-generation sequencing. And this is another type of revolution. We have gene expression omnibus. We can store all the arrays. Fantastic. Then, oh, sorry for this, but this is basically Illumina sequencing. And now we can profile short sequences of nucleotides massively, right? And one example of this project, I mean, we can do whole genomes and not in 20 years, but in X number of days. And one of the first projects that is really worth mentioning is these 1,000 genome projects, right? Where they say, well, let's do the whole genome for 1,000 individuals. What happens? Storing 1,000 genomes is not easy. Then you need to start having teras of resource, I mean, of storage, and how you store it and how you create it, etc. So one idea could be, okay, let's have another type of resource, because in macro arrays, we are talking about megas of information. Now we are talking about gigas, terabytes. So we need to compress the data. We need to make better ways. So there was another generation of, let's say, toolkits and ways of storing, sharing, etc. And again, this international consortium for storing all this information. Fantastic. But why do you think right now we are not allowed by any means to share sequencing data, human sequencing data, in gene expression omnibus? The reason came, I mean, and I remember when I saw this presentation in Boston, they took the 1,000 genome project and they set themselves a challenge. We are going to use the 1,000 genome information to identify the individuals. And they made it happen. For 50 individuals, they were able to find them. How? Well, you can go into the paper. They use these 23andMe databases, surnames, different constraints, etc. They were able to identify them. And that makes a game changer, because if you can de-identify the individuals through the genomic data, then we have a problem with anonymization. So that was one of the reasons that I think many started to think, okay, gene expression omnibus is not enough. Those type of resources are not enough. We need to have other type of environments. And this affects directly decision, right? So one of them is the dbGaP, database of genotype phenotypes, where it's developed to store all this information of genotype and phenotype in humans. You can see this is the statistics from the website from a few days ago. We have the European kind of analogy one, aiming for the same things. And how it works is basically you want to have an environment where every one of the researchers can identify those data sets do exist. And if you identify them and you think they are relevant for you, you can ask to have their access, right? So this is exactly how it works. You submit it, fine, you still have full control, and then you have some data access committee, so any researcher can identify it and say, well, this is interesting for me, please let me access. Then it's evaluated, and then you take responsibility of the data. So that it's important for many of the data that we are talking about. Any sequencing data is affected by that. Any sequencing data that goes to this aspect. Now, there are other type of projects that I think also has its own space here, because the TCGA, the Cancer Genome Atlas, it's something that is amazing because they went for many cancers, thousands of individuals, hundreds, thousands of individuals, different omics. So they were profiling for some of the things they were profiling, like mutation, copy number, gene expression, etc. And they say, well, it's not enough to have So they created their own repository. Initially, it was possible to access and download the data. Now you have, again, a data access control. You have to say, well, this is my institution. There is a data controller. All these ethical aspects that we have been discussing in the project at certain times. Now, another thing is, in this challenge of sharing the data, is a question that comes to us saying, is sharing data just enough? In 2001, you have several papers on Nature Review Neuroscience talking about, okay, if you are sharing the data, the quality of the macroarray data is going to matter a lot, right? And they make a collection of how it was accessible through websites, it was accessible to different resources, even companies were commercially making available some databases. And then there are other, I mean, it's as close to the raw data. Please do not share the process data, because maybe the way you process the data 10 years later is not the right way. So you want to have the raw data, so you start from the beginning. And it was interesting because at some point, it was aiming to enforce how, well, a suggestion, this is the Miami recommendation, how the data has to be shared, what information has to be given, how. I'm going to show you here. This is what information in the Miami standard it's show there. 2006, discussing about it, right? It's a bit later. And again, we have the, let's say, the discussion, is this a guideline or is this a standard? Guideline means it's a suggestion. The standard means it's enforced. And the point here is finally, and it took time, it became enforced. And then it has been evolving over the time that we also have the fair guiding principles for sharing all the data that we generate, right? How to make it useful for everyone in the future. For closing this section, I wanted to show a few things that I think matter. So the future of the database is also going to be evolved, because you can think about the DeepMind AlphaFold, where now they do this 3D conformation of the proteins. AlphaFold, 3D conformation of a protein was extreme challenge. And the resolution was 20, 30%. Then came AlphaFold and say 60. And then AlphaFold to 90. And then it's like, it's not a solved issue, but then they are creating, and then with the AMBL, they have this kind of updated database. And many things, I think, may move in this direction if we have many automatic tools. Another aspect that I will think, and this is my final slides, is we are talking about maybe gene expression, DNA methylation, etc. But databases are growing to another level of complexity. Like right now we have a spatial transcriptomics, right? The 2D dimension of the expression over a layer. Now we are having papers where you use these 2D dimensions to reconstruct the 3D expression levels of certain tissues, organs, etc. Can we start storing this information in an organized manner, in a fair principle-based manner, so everyone can access it? So I think our challenges are within decision is not here, but this is what is coming. So with this, I tried to give like an historical perspective. Hope it was useful, but then we go more practical within the decision.
Hello. Well, I will not talk so much because I really prefer that Ferran going deep on what is really going on with the databases we have already created in Decision. But what I would like to explain is when I started here as a work package 2, because first, at the very beginning of the project, there was another guy, another girl. I really don't know what fair principles mean. No, it's like, okay, you have to integrate the clinical database, the omics, well, the omics database together with Ferran. But then, in fact, these fair principles means GDPR, means how to share data, means bioethics because of the identification of patients and a lot of things, no? So, and all this kind of information was a deliverable for decision, the data management plan. So, and it's really linked with the FAIR principles, no? Is how to FAIR DF is find the data. It's okay, find the data, but you have to define the format, the origin of that data, where are you sending this data, in which format are you sending this data, how is it stored, because it's like it's not whatever server you can upload the data. And then how to access is like all the people can access to this. Yes, because it is supposed that this should be reused, but not whatever person can apply or can do whatever they want with the data. No, the integration is like, okay, you have the clinical data or the omics data, but what you were saying, no, is like you have to put the raw data, because maybe if you upload the process data, is useless for the rest of the people. And then, well, optimize the further use. Now is another of the deliverables was to have a data hub for decision in order to make reusable for all of you and even people outside the decision consortia this data. Also in the data management plan, even we have how to publish and how, well, the publications, they should be also open access for all the people. So this is something that you have to keep in mind. Okay, so where are the data of decision? So it is in the data hub. In here we have all the omics of decision, like microRNA, the methylation data, lipidomics, metabolomics, extracellular vesicles, all the omics are here, and also the clinical data. Genomic data and transcriptomic data is what you were explaining before in an archive. Also, it was according to the decision project, it was a deliverable to upload all this data in archive. So if these are the accession numbers. So if you write this in a browser, you will find, well, the decision who upload this, a little bit explanation of what is uploaded. So, and what you were explaining before, if someone really thinks that this could be used for the project, they can apply for the data, and then there is a process for approval this data. Which is the limitations of the archive? Limited clinical data. What does it mean this? Right now you have all the sequencing, all the genomic data, but in terms of clinical data, what you have is an autonumeric, like 1, 2, 3, and very few data. If I remember well, Ferran was like, it was an ID, pre-CLF, and not so much, sex maybe? Yeah, so if you want to go in deep in this data, the ideal situation is to go and to ask for the data behind of this. No ID patient. So for example, if you say, well, but I know the ID patients because I work on the decision data. So you are not able to identify this if you don't ask to ask for this correlation of data. And then this is also important under embargo. We are so close to, well, in fact, once we really publish the paper of Richard and the other one of genomics, we will open this embargo. In fact, the general rule is once the first core paper is published, this will be open for the rest of applicants. If you want to have all the specifications, they are in the grant agreement also specified. Well, here you have the access grant. And right now in the data management plan, there are like 3 sorts of sharing the data. So if you are like a work package leader, task leader, meaning that you have analyzed any uniomic data of work package 1, and you want to do an extra analysis related to a task of decision, let's say is like the easy, no? Because it's like decision, people from decision, a task from decision. So it will be almost direct to share the data with you. Then we have another step is like, okay, I am from decision, but I wanted to do a step beyond. So then we have like a kind of steering committee, people from, well, Pierre Emmanuel is there for deciding if we can share the data or if it is feasible or not. And then finally, we have like the third step, that is people outside decision that wants to use data from decision. Right now, we don't have yet this kind of petition, mostly because it's like we are waiting for all these kind of publications and core papers from decision. Well, you will receive some kind of email like that. And then once you have your user and password for accessing to our data hub, what you receive is, well, you access this. This is an overview per role and with expiration date. This is something that should be clear. When we open our data hub, is always a partial information of what you have asked. You don't access to all the data we have uploaded, has an expiration date. So we have found sometimes people that says, no, the password does not work. And it is like, no, you spend like a couple of months on downloading the data. So it expired, this password. And you can find several types of data, like Excel files, CSV, SAS data, and then the worst, let's say, part, and right now I think in this room only was Joanna, the one who see this kind of a screenshot, is the RNA and the genomic data. So since the size of these files are very heavy, we have a special server for that with an special access, because what we do is not copy and duplicate information, but to do let's say a compartment of that information, and a password for downloading this. And this sometimes is so huge. It depends on the connection. It depends on the servers of the people who is accessing. Could be the very worst part of downloading data, but people do. And now, as I said before, Ferran will explain about the cooking.
Oh, well, my goal with this presentation was to try to teach something about the bioinformatics and the access to the data, and more how to access. Like, I wanted to give an overview of what to expect when you get the hands on the omics data, because we have the feeling that when people are applying for to access to our data in this project and other projects, they don't really know what they're getting into. No, they apply for transcriptomics, they apply for genomics or whatever, but then they receive the data and they don't know what to do with it. So I wanted to use this masterclass to give an overview of like every omic, and to teach you what to expect when you download this data and what to be ready for it. So when we were discussing with Christina this presentation, we came up with the idea of talking about this like a mise en place in a restaurant. No, like okay, I know what I want to cook. Like my objective is to cook this plate, but first you need to have ready all the ingredients. So the first thing you need is a well-defined question. This may sound silly, but I think it's the most important thing, because a lot of people ask for data without knowing which is the question they want to answer. And when they receive the data, they don't know how to start. So I think the recipe is like really, really important. Then the tools, you need proper software, proper hardware. Software would be the tools like a server where to analyze the data, and the hardware would be like this to store all this data and all this information. Next would be all the ingredients. Another problem we find, maybe because people don't know what to want to achieve with the data they are asking for. We have several rounds of clinicians or statisticians asking for more and more data because they don't ask at the beginning for the whole thing that they would need, or even they ask for more things that they need and then they get confused and overwhelmed with the amount of data they received. So I think it's pretty important to know exactly what you need. Also, knowledge on how to analyze this data, or having access to someone that knows how to do it. I think nowadays with AI and all the tools that are around there, you don't need to be a top bioinformatician to get interesting results. Maybe I'm shooting myself in the feet now, but I think you need an expert to help you and guide you and be sure that you're not making any mistakes or analyzing things in a wrong way. But I think nowadays and in the future will be more accessible to anyone. And finally, time to cook this all this data and to get proper results, because we feel like people sometimes are not ready to invest all the time needed to get the results. Like you have physicians that have an idea, ask for the data, and time passes, and obviously they don't find time to work with this data. No? So now I try to go over all the omics data we have in Decision to answer all these points really quickly. I mean, obviously, this is my experience. I'm sure there's a lot of ways to analyze this data and to integrate them. I'm not teaching how to analyze the data, but just doing an overview of what to expect. So I will go from easiest to maybe the hardest one. First, we have like omics data is similar to clinical data. In this case, we have cytokines, RNA proteomics, targeted metabolomics, and lipidomics. In this case, it's you can receive this data in an Excel file, so you don't need a big server to analyze it. You can analyze it with basic softwares, PSS. I don't like that, but you don't need to know programming. You just need basic statistical skills. Normally, you need to pre-process this data to adjust for the correct distributions and so on, but it's not a big deal. Yeah, and some of these features, you need some previous information like limits of detection, like for metabolomics and cytokines, and to know which are the ways of dealing with these limits of detection. There's several approaches. You can find it somewhere, but most of them are like very easy to apply. So next, on targeted metabolomics, I think this is a very interesting data we have in Decision. It would be a pity that people is not applying because they don't know how to access them. In this case, you have a huge number of peaks. Maybe you receive this data in a text file or an Excel file, but it's a huge amount of information. You also need to log transform this data. The limit of detection, again, you need information about that and how to apply it to your data. Also, it's very important annotation of the HMDP codes, just in case you need to do some enrichment analysis. So to use all the websites around there that help you to do it interactively, you need these codes properly annotated. So for this, you would need either to ask in the EF CLIF, because we have these codes, or to the platform in charge of the performing the analysis. Also, the batch effect correction, we have during many years, we discussed with the platform because this is real data. This is EF CLIF data. In this case, you have 4 batches. One is canonic, one is Aclara, the other 2 Aclara canonic predict, and the other I think it's another batch of Aclara or another batch of predict. For many years, we thought it was not possible to correct this batch effect, but this year I proposed myself to solve this problem, and I managed. But it's an issue that can be solved, but you have to do this with different packages of R. You can do it with Python or whatever. And finally, the analysis, you can use basic fault change computation between groups, adjusting by covariables, and then performing enrichment analysis. This example of a volcano plot, the enrichment analysis you can perform. This can be done in websites. Transcriptomics, this is from our last presentation. In this case, you need a server with enough capacity to store this data. Many people apply for this data, then they realize they don't have a lab, but you cannot store this on your laptop. We think most people expect us to send already processed data, but this is not as easy as it may seem, because to process all this transcriptomic data, you take like 30, 60 minutes per sample. You can do parallelization, but more or less like that. And all these steps include the filtering, aligning, quantifying, and normalizing the data. Problem with this is that when you add new patients, imagine someone applies for 80 patients, but later on, 2 weeks later, they receive the data, and then they realize they also want to include ACLF patients. Then you have to perform all this normalization again. So it really takes a lot of time. After all this, you just get the normalized count, and you can start working, and you can perform again fault change computation, enrichment, WCNA, all the kind of analysis. And finally, genomics. I wouldn't say is the most difficult, but it's one of the more complex. In this case, the raw files are IDAT formats. You have the red. You have to preprocess this data with Genome Studio if it's in Illumina. It's our case. And for this step, you really need help from the genotyping platform, because you need to know which chips they used, which version, which year. In our case, I think we have several versions of different chips used. So really a bit messy. After that, you obtain files that you can process in a software that's called Plink, can be also accessed by R. But normally you cannot do this with other platforms. And with this, you can perform quality controls, filtering, and all the genomic analysis. And I think that's all. You have questions?